Greetings, students, and welcome to this, our last series of screencasts in the unit on genetics for AP Biology. And these screencasts are going to be about DNA technology. So, as you are probably aware, humans have actually learned how to manipulate DNA in a lot of different ways. And so, in this unit, we're basically going to look at how. And along the way, we'll kind of take a peek at some of the ethical issues that arise with these new advances. So the first technique I want to look at is something called recombinant DNA. Recombinant DNA is simply a molecule that contains DNA from two different sources. In other words, an example of this might be if you inserted a human gene inside a bacterial plasmid, and we're going to look later at why that might be an interesting thing to try. This works, remember, because the genetic code is universal, meaning that if a piece of DNA finds itself in a bacterial cell, it will be transcribed and translated in the exact same way to form the exact same protein as it would if it found itself in a human cell. We've alluded to this a little bit before. If you recall, we talked about how you can take the gene that causes bioluminescence in a jellyfish, put it into a cat embryo, and then that protein will be expressed in the cat and cause it to sort of glow, which is awesome. Or the firefly gene, that protein that causes the firefly light to blink can be put into a tobacco plant or a pig and cause them to light up, so to speak. So how do you do this and why would you want to? So first of all, how do you do it? Well, we're going to break it down by players and process. So the first player are these enzymes called restriction enzymes. And what a restriction enzyme does is it cuts the DNA at a very specific site, which is called a restriction site. And when it does that, it produces what are called restriction fragments. So imagine you've got a stretch of DNA here, and it turns out that this particular sequence, CTTAAG, that is a restriction site. Okay, and you'll notice it's the same backwards here, CTTAAG. And so this is a location recognized by a particular restriction enzyme. It's going to bind and it's going to cut the DNA at that specific location. And here's another place where that particular sequence is, so it's going to cut it there. And the result is that you get what are called restriction fragments, these little chunks of DNA, so to speak. Now, I think we've alluded to these before, but it was very brief and minor. Where do we find these in nature? These are actually found in bacterial cells. It turns out that bacteria use these restriction enzymes as a defense against viruses. So if a virus injects its DNA into a bacterial cell, this cell has restriction enzymes that can chop that viral DNA up and restrict its activity, hence the name restriction enzymes. They restrict the virus's ability to replicate itself inside that bacterial cell. So we chop it up, we get these fragments. Now you'll notice that these little fragments have these single-stranded ends on them. Those ends are called sticky ends. Why are there these sticky ends? Why are they called sticky? What makes them stick? Well, remember, these ends, these single-stranded portions, will hydrogen bond to complementary bases. And so since they're unpaired at this point, they want to hydrogen bond with a partner, and so we sort of call them sticky because they tend to hydrogen bond with other things. Our next player is DNA ligase. DNA ligase, that sounds familiar. Where did we see that enzyme again? DNA replication. So when we talked about DNA replication, we talked about how the lagging strand is produced as this series of Okazaki fragments. And so DNA polymerase travels along 5' to 3', always adding nucleotides in the 3' direction. When it catches up to the primer, that RNA primer from the next Okazaki fragment, it will actually remove the RNA bases and replace them with DNA bases. But then when it gets to the very end, that last little place, there's a little gap there that it can't quite fix. And that's where DNA ligase came in. DNA ligase connected the sugar phosphate backbone here with a covalent bond and just kind of sealed that little gap and joined the Okazaki fragments together. So we're going to use DNA ligase in this as well. Our next player is a vector, and a vector is simply an agent that introduces recombinant DNA into a host cell. Oftentimes we use either a plasmid or a virus to do this. A plasmid, you may remember, is those little extra rings of DNA. So a bacterial cell has its main circular chromosome and then these little extra rings of DNA. That's what a plasmid is, these little extra rings. Now, in order to be effective, a vector must contain an origin of replication and a promoter. This makes sure that that vector DNA can replicate itself and be transcribed and translated. It needs restriction sites so that it can also be cut with restriction enzymes. We'll see why soon. And then it needs a selectable marker. This is a gene that causes an easily identifiable phenotype. So an example might be resistance to an antibiotic. So AMP-R gene means resistance to the ampicillin antibiotic. This is so we can find it later, as we'll see. The final player then is the host cell. The host cell is simply going to replicate and express our recombinant DNA. Oftentimes it's bacteria. It doesn't have to be. It could be yeast or even some mammals. But we need something that's going to take that recombinant DNA and actually make the protein for us. So how does this all break down? Instead of modeling this now, I think I'm going to go through the slides and then I'll cut to another scene and we'll model it in summary. So the first step in the process is that the DNA with the gene of interest and the DNA of the vector are going to be cut with the same restriction enzyme. So let's suppose we wanted to take the human gene for insulin and put it inside a bacterial cell. Okay, that would allow the bacteria to produce insulin for us. So we take the human DNA and we chop it with a particular restriction enzyme like BAMH1. And then we take the plasmid from a bacterial cell and cut it with the same restriction enzyme and cleave that. So we've got a human gene with sticky ends. We've got a plasmid with sticky ends. Well, what's going to happen next? Those sticky ends, being sticky, are going to hydrogen bond to one another. And when they hydrogen bond to one another, that is going to link the DNA from those two sources together. And so it's a little subtle, but you can see this darker insulin gene within the lighter colored plasmid. Note that some plasmids do not take up the gene and just seal back together again. And so, that's something that has to be dealt with, and ignore that far right pick because that was a different picture. Forgot to edit that. Oops. Once they're sealed together, then DNA ligase is going to join the sugar phosphate backbones covalently and kind of seal this up. So now this is sort of one single little piece of DNA. Then what happens is that vector is going to be taken, here's our vector, into a host cell, and that is going to be allowed to replicate. How do we get it in the host cell? It could be taken up by transformation, which we learned about in our last chapter. It could be injected by a virus. There's lots of different ways to get it in there, but it's going to be put inside that host cell. And then the host cells are going to start to grow. And when they grow, they're going to divide and they're going to copy their DNA. And when they copy their DNA, they are also going to copy this plasmid, and they're also going to copy that gene of interest for us. Okay. Now, the host cells are screened with a selectable marker. In other words, when we're growing these things, we don't just grow them on any old surface. We grow them in a way that allows us to make sure that they've actually taken in that vector. So in this case, we might have this ampicillin resistance gene in that plasmid. And then if we grow the bacteria in the presence of ampicillin, only cells that have taken up the plasmid will be resistant, and only they will be able to grow. So that way we know that our cells actually took up the gene. So at this point, all we need to do is keep our bacterial cells growing, and they're going to produce human insulin for us. Slick. Here's just a little recap, a little nicer picture. Take the human insulin gene, we splice it into a bacterial plasmid, put it into the bacterial cell, and then we grow it and extract the insulin. So applications of this process, well, we've already talked about this. Human insulin production, right? We can now grow bacteria that are able to produce human insulin for us. And then that insulin is used for people with diabetes. In the past, they had to isolate that insulin from pig pancreas, which was pig insulin and had to do with pigs and pancreas and that. So now you just need a vat of bacteria, Easy to grow. They'll crank it out forever. Easy to maintain. It's beautiful. Um, Another application is in producing transgenic organisms. These are organisms that contain a gene from another species that has been inserted into their genome. So, an example of this would be farming. That's a PH farm. That's using farm animals to produce medically useful products. So, an example, there is a gene for a blood clotting protein. This is given to people with hemophilia. When you get cut, blood leaks out of your opening, but then hopefully it clots, which means it kind of clogs up the wound and the blood stops flowing. Well, if you have hemophilia, you don't have that clotting protein, so your blood just keeps going, and that's a real problem. So they need this medication. Well, that gene for the protein that does that is actually placed into the genome of a goat. That protein is then produced in the goat's milk. And since goats make a lot of milk, they make a lot of protein that we can then isolate from that. So it's a way to get a lot of this protein that can be then harvested for medicine. Another transgenic organism, plants that make their own insecticide. BT corn is an example of this. Odds are good you've eaten plenty of BT corn in your life. The BT gene is produced by a parasitic bacteria that lives in the guts of insects, and it produces a toxin that actually kills the larva of those insects. So scientists took that gene that kills these insects, and they put it inside a plasmid, and they put that inside corn cells. So now the corn is actually making the protein that will kill their own parasites. And that makes the corn resistant to the insects, which helps us get more corn out of the deal. Um, we can make plants with improved nutritional traits. So golden rice is an example of this. We're still working on this one, but there's a lot of people who are deficient in vitamin A. So some clever person thought, hey, what if we splice the gene for vitamin A production into the rice that many of these people live on? And so by putting that gene inside the rice plants, then it can produce vitamin A is kind of, it gives carrots its orange color. So it produces what's called golden rice, and we can end up helping these people prevent the problems associated with vitamin A deficiency. We can make plants that thrive in unusual conditions. So, you know, a gene that allows plant cells to transport salt into their vacuole and store it was put into crop plants, which allows them to be raised in poor quality soil. So here's the plants in like a really salty area, but if we give them that gene, here's that same plant. And so this again helps you know, with climate change and changing conditions, it can help ensure that people have the crops they need to stay alive. We can make bacteria to clean up environmental problems. So there are bacteria that have genes that help break down harmful chemicals like oil spill stuff or metals from mining. And so we can actually, you know, those bacteria aren't always in the place in the world where we want them. So we can take the genes from those bacteria and then put them in bacteria that are native to an area of an oil spill. And then those genetically engineered bacteria in this area are going to slowly break down that oil spill and help prevent that oil spill from being even more destructive than it already is. So there's lots of applications of this, and it's already happening. And you've probably heard a little bit about some of the controversy. You know, GMOs, that stands for genetically modified organisms, which is what we're talking about here. So anything you've heard about GMOs, that's this. And so you know, the reality is that the US produces and consumes huge numbers of GMOs. Odds are good you've eaten this. You know, 88% of our corn is GMO corn, for good or ill. Advocates of this will say that genetic manipulation has always been a part of agriculture. And so, you know, by selectively breeding, we're sort of tinkering with genetics anyway. Most genes inserted are very specific genes, specific to a plant or bacteria, and therefore, in theory, shouldn't affect humans. And then it can help us with a lot of problems the world is facing. And we've seen this in examples, production of medicine through insulin and the blood clotting protein, malnutrition with that golden rice, allowing plants to grow in changing climatic conditions, climate condition, climactic, that's the wrong word, and then cleaning up environmental issues. Dissenters say that, well, this is different than just artificially breeding certain traits. This is definitely unnatural. They suggest that foods may not be safe to eat, There isn't much research supporting this yet, but that doesn't mean it's not true. And then genes or whole organisms can escape and then appear in other in species, which can be disruptive, and we have seen that happening. So we're out of